This is a proposed hypothesis to explain the pathophysiology of age-related bone loss and senile osteoporosis. The young bone marrow is a very active milieu in which osteoblasts, seen here as purple cells, share the marrow space with adipocytes, which you could see in yellow, osteoclasts, and mesenchymal stem cells. The key players in the process of aging bone are known as mesenchymal stem cells. These cells have the capacity to differentiate into either adipocytes or osteoblasts. This differentiation will depend upon the presence of specific growth factors and hormones, as well as the expression of transcription factors. In the young bone, there is a predominant differentiation of mesenchymal stem cells into osteoblasts, although some of them differentiate also into adipocytes. So at the end, what we have is stem cells differentiating into either adipocytes or osteoblasts, osteoblasts able to build up new bone matrix, and adipocytes there with a so far unknown function in terms of bone metabolism. So let's talk now about the characteristics of old bone. In the old bone marrow, you could notice the predominant presence of adipocytes, seen here as yellow cells again. And this prevalent presence of adipocytes may determine their close interaction with both osteoclasts and osteoblasts. A summary of changes in cell population in the aging bone is illustrated here, where we find high levels of osteoclast differentiation and activity, low levels of osteoblastogenesis and bone formation, and high levels of adipogenesis. There is increasing evidence demonstrating that adipocytes exert a lipotoxic effect within the bone marrow. This lipotoxic effect is explained by the secretion of factors, including adipokines and fatty acids, that affect osteoblastogenesis and bone formation, induce osteoblast apoptosis, and activate osteoclast differentiation and activity, with subsequent bone loss, increased bone resorption, and osteoporosis.